Well, hello and welcome to our Phytophthora Crop Protection Network seminar. I'm excited today to be sharing the presentation with the former PhD student, now Dr. Austin McCoy. And what we're going to talk about is taking the headache out of managing Phytophthora root rot in soybean. I'm really excited to have Austin here because he actually did his PhD on this particular project. And so we've got a resident expert with us here today. So let's get started. Just in general, this is what we're going to cover today. We're going to give you some of the behind the scenes work that Austin did to try and address some of the management concerns we have in Phytophthora. So we'll go through, talk about some of the RPS genes for management of Phytophthora, what they are. We'll talk about partial resistance or field tolerance, as it's also known. Some fungicide resistance work that Austin did, and then some really exciting work that he's currently looking at in terms of assessing these RPS genes, not just in the US, but globally. So let's keep rolling. So Austin, would you like to give us an overview of the Phytophthora sojay disease cycle?
Yeah, thanks, Marty. So I'll be starting over on the far left with the dormant oospores that survive in the soil. So these oospores are the resting structure for Phytophthora and Pythium species, and they can survive in the soil for many years. And they wait there until there's an environment which is conducive for disease. So flooding saturated soils that allow for pooling and free water to be available. Those oospores will germinate either into hyphae or into what's called a sporangia, which contains many of these small motile spores called zoospores. And they have small flagella or tails on them that allow them to swim through water. And they're attracted to soybean roots. So they will swim towards the soybean roots and attach themselves and then start infecting and colonizing those roots. During that infection and colonization process, you'll get the formation of new oospores, as well as starting to get some of those symptoms that we
Typically
starting
see in the field, right? I mean,
right.
so this picture on the right-hand side is a great example of that, wouldn't you say, Austin?
Yeah. So we start getting some of those root rots and eventually the stem canker, wilting, and chlorosis of the plant towards the end of the season. Well, it can be during any time of the season, but we typically see those stem cankers mid to later season. And all of the oospores that were formed in that year in the roots or stem of the plant are then put back into the soil where during tillage, or if we leave stubble, it can reside there for quite a few years.
Yeah,
And on the
absolutely.
right is our diagnostic feature for Phytophthora stem and root rot. It's this nice chocolatey brown lesion that starts at the crown of the plant and works its way up. And it's very easy to diagnose in the field.
Mhm. Absolutely. So we're looking, if we're trying to diagnose this in the field, we're looking for that initiating from the soil line and moving up, right?
Right. There can be some stem cankers that can look similar. So, like Diaporthy stem canker can also cause some death of the above ground stems of the plant. But those typically start off at like where a petiole attaches to
Mhm.
the stem or where a branch attaches to the stem, not coming up from the soil.
Yeah. Yeah, so that's a really good indication if you're trying to identify Phytophthora root and stem rot. Yeah, like Austin said too, I mean, it's certainly, this disease is certainly capable of killing seedlings, and in that situation, you might not get this what we call a classic sort of, Phytophthora symptoms. So you may still need additional diagnostics to identify things, but certainly a great characteristic to look for in the field to help identify this particular disease. And just by way of additional explanation too, so we're looking at cross sections of roots down here as well. So you're looking at that infection process and those little oospores forming in the root that continue into the soil. So
We have a few different management practices that we can do to try and limit the disease pressure and incidence in the field. We have genetic resistance in two forms. We have qualitative resistance. These are those RPS genes that protect the plant from any infection and disease if they're effective. And in the U.S., we typically see the main some of our soybean varieties contain RPS1C and RPS1K, with some varieties containing RPS1A, 3A, and 6. And 1A, 3A, and 6 are not very common in the seed treatment or the seed catalogs that we've looked through. We also have quantitative resistance, which you may know as partial resistance or referred to as tolerance. This is not the same as qualitative resistance. It is likely many genes acting together that just provides a tolerance for the plant to the pathogen. If you're looking at a seed catalog over to the right, you can see we've highlighted in red the Phytophthora resistance genes that we were talking about for qualitative resistance. And then in green, we've highlighted the Phytophthora field tolerance score from this particular vendor, which would be our quantitative resistance. We also have some seed treatments that are effective against Phytophthora sojei. Mefenoxam or metalaxyl, which is a FRAC 4 found in apron-like products. Ethoboxam is a somewhat newer chemistry. It's just been out for a couple of years now. These are in the Intego products or FRAC 22. Our newest compound to control Phytophthora sojei is oxythiopyprolin, which is found in the Lumicina seed treatment and is FRAC 49. And then there's been some evidence to show that some of the general fungicides, the QOI fungicides like pyraclostrobin, also have efficacy against Phytophthora sojei when used as a seed treatment. Besides genetic resistance and seed treatments, if you have areas of the field that are reliably pooling water every year when it rains, you can utilize drain tiles in some of those poorly draining areas. Or if you've had to do a lot of passes through your field and the soil is very compacted, you could look to potentially tilling to try and reduce some of that compaction and allow for better drainage of the soil.
Excellent. All right. And so this Austin, this was your PhD project, right? Where you did work looking at a pathotype survey. We should talk about that briefly, right? Like some people may be familiar with the race system of Phytophthora sojei, and now we've moved to pathotype. Do you want to just tell us what that's about, Austin, just briefly?
Yeah. So previously we've been referring to Phytophthora sojei by races, which a race would determine which resistance genes were effective against the pathogen. We've started to transition away from that towards referring to isolates of Phytophthora sojei by pathotype. And that is just a list of resistance genes that the isolate can overcome so that it's a lot easier to talk about with folks and we can identify, what resistance gene do you have in your field? What pathotypes are in your field? Is that resistance gene going to be effective for disease management? So the race versus pathotype, they're both describing similar things in different ways. And we just feel that pathotype is much more translatable to everyone instead of race.
And so what did you do with, this is sort of some of the main areas of your PhD work. So what did you do here in Michigan?
So here in Michigan, we collected soils from as many counties as we could sample. And we used those soils to bait out Phytophthora sojei and test it against, I believe, 13 different resistance genes. So you can see here, this is where we collected soil from and counties which were and were not positive for Phytophthora sojei. Once we had those soils, we were able to bait them using a baiting process where we essentially produce a conducive environment for disease. So we take that soil, we homogenize it so it's very fine, put it into cups, and flood them so that they are very moist. We then bag them for a couple weeks to allow for that humid and moist soil condition to continue, which allows the oospores from Phytophthora sojei to start germinating and primed for infection. We then planted those soil samples with a variety of soybean that is just completely susceptible. There's no resistance gene in its genome. And we use those plants to bait out the pathogen. So we again flood the fields so that those motile zoospores will be attracted to the roots. And you see here where Marty is circling the diseased seedlings that we were able to take out and perform isolations on to obtain pure cultures of Phytophthora sojei for testing.
And this is quite a labor-intensive effort, right?
Oh yeah. I mean, it was certainly not quick and not easy. I mean, once we got the 5-gallon bucket full of soil, we took it and put it through a soil mill, essentially, just grinding up all the dirt clods, making sure it's all very fine and mixed together. I mean, it would take close to a month between having the soil and getting isolates out of the soil so that we could actually do the work that we wanted to do, right?
Yeah.
This is all just prep work.
Yeah, just all this work. So I just want people to appreciate, that it does take time. There are some other really exciting developments in terms of molecular markers. There's a Canadian group that are really pushing along on that, and so that's really neat. Maybe we'll have some other faster technologies to try and get some of this sort of information that we're talking about today. So what did you do next? So you had this big collection of Phytophthora sojei isolates that took a lot of effort to collect. Then how did you assess them?
We took those isolates and we used them to inoculate 14 different soybean cultivars. And each one of those cultivars, which we referred to as a differential, they each contain a single RPS gene. So a single gene used for management that could be used for management of Phytophthora sojei. And we inoculate them with Phytophthora sojei and then record, was the plant diseased? Was it not diseased? What was the severity of disease? And you can see Marty's pointing towards the rating scale on the right, where on the far left, we have a resistant soybean line. You really can't barely tell that they were inoculated by the time we do the ratings, as compared to some of the lines that were not resistant. You get a variety of lesion lengths depending on the isolate and the soybean variety.
Mm-hmm.
And so through this, we were able to say, hey, these isolates can cause disease on these genes, but not the other genes. So what we're seeing here is a cartoon representation of that, where you can see in the soil line here are the different RPS genes that we tested. On the far left is a susceptible control that has no RPS gene. And post-inoculation, we would go through and count the number of dead or dying plants for each soybean cultivar, tally it up, and see which ones were resistant and which ones were susceptible. Mm-hmm. And this is quite literally what it looks like. If the soybean lines were not resistant, most of the time when I went back a week later to rate them, they were just completely dead, and there were only a few lines in our testing arrangement that were still alive.
Mm-hmm. And this as well, I mean, we talked about the amount of effort to collect the isolates. That does take time and effort. And then this procedure here, Austin may have a Band-Aid on his thumb. It takes quite a bit of effort. And
so you can see in the top right how I did this. I used a hypodermic needle and a syringe, and for every one isolate of Phytophthora sojei, approximately 140 soybean seedlings were inoculated individually. So we couldn't just inoculate the whole thing real quick. I had to go through and individually inoculate each plant for testing. So yeah, very time intensive.
So I really like this cartoon, and just to emphasize one more time, so this is a single Phytophthora sojei isolate, right? So this is a different pathogen, but a single isolate of Phytophthora sojei. So there might be multiple different types out in the field, but this is one of them, and this is an example of the reaction type. So for this particular isolate, we can tell that only the 3A, 3C, and 4 gene were effective in managing this particular pathogen. The others were not effective. And so this is where the information is really, really valuable, I guess, to know which genes are actually working out there in the field. So we had a resource as well, Austin, didn't we? So, so 20 years ago, a very similar study was done here in Michigan. And a lot of this information we're talking about too applies to other states, and we'll get to that at the end of this presentation. But about 20 years ago, in 2001, they published this study looking at, again, which genes are effective at managing Phytophthora sojei isolates. And I'll just point out here on the scale, we have that soybean differential that has no RPS genes in it. So it's our control isolate. So we expect 100% of those plants to die. And if they don't, we know something's wrong with our test and we need to redo it. But this is essentially what they found last year. So the higher the bar, the worse it is for that particular gene. And we use this sort of artificial cutoff of 40%, right? So 40%, if more than 40% of isolates are able to overcome that particular gene, then it's no longer effective. So in this particular instance, we had 1C. It's still effective for about 48% of isolates, but it's just over our threshold. So, it's not sort of effective as we would like. And we've circled in blue there isolates that we were below that threshold, such as 1B, 1K, that were 20 years ago effective in managing most of the population. So 1K was able to manage about 70% of the isolates out there. There was only about 30% that are able to overcome 1K, and similar sorts of numbers with 3A and 3B and 6. So you put all this effort in, Austin, right, to have a look at it again. And we didn't know what we were going to find here in Michigan. And I mean, the results were really very interesting. And Austin, why don't you walk us through what you found?
Yeah, so like Marty said, 20 years ago, it appeared that RPS1B, RPS1K, RPS3A, 3B, and 6 were effective. What we're seeing now is that not only are those resistance genes not efficacious anymore, but the RPS1 loci, so RPS1A, 1B, 1C, 1D, and 1K, those are all now 80% or more of the population can overcome those resistance genes, which is very problematic because, like we've talked about, RPS1C and RPS1K are our two most prevalent resistance genes that are available. Um, there were genes like RPS3A that didn't change in efficacy too much, and could still potentially be used as an effective means of management. But we were very surprised to see this level of the percentage of isolates able to overcome these resistance genes. I mean, 80% plus were able to overcome RPS1C and RPS1K.
And so you did some additional work as well. And again, this is a little bit behind the scenes to look at this data, right? Do you want to explain what we're looking at here, I guess?
Yeah. So in addition to looking at which RPS genes were efficacious in Michigan, we also looked on the pathogen side to see how many resistance genes could these isolates overcome. And this was also done 20 years ago as well. And you can see 20 years ago, we had a relatively even distribution of pathotypes. There were a couple humps there. But on average, an isolate could cause disease on 7 RPS genes. When we went back and did this for our study 20 years later, we see that those pathotype complexities, or the number of RPS genes an isolate can overcome, has shifted to the right. And on average, isolates now are able to cause disease on 9 RPS genes. So that's 2 additional genes in 20 years on average that isolates can cause disease on.
Yeah. So what you're saying is that a single isolate can potentially overcome more RPS genes than 20 years ago.
Yeah. On average, isolates now can overcome 2 additional RPS genes as they could 20 years ago. Which is going to make it very difficult to manage Phytophthora sojei going forward with just single resistance genes in soybean germplasm.
Right. Oh, there's your means anyway at the bottom there. 7.2 to down 9.2. I really like this particular image. So what we've been stressing at the moment has been those RPS genes, and they're more over black and white. You know, you either have
Yeah.
it working, right? Yeah, you agree, and
Yep.
you have resistance, or you don't, and it's failed. This particular example I like because it also talks about the value of that, what we call tolerance, and that's typically what you see in the field catalog. Sometimes as pathologists, we may prefer partial resistance, but anyway, regardless, this field tolerance idea. So what was really striking about this is that the farmer had split the planter with two different varieties from the same seed company. Both of the varieties, the soybean varieties, contain the RPS1K gene, which Austin had found, unfortunately, is pretty much not very effective here in Michigan, with about 80-90% of the strains of Phytophthora out there capable of overcoming, right? And you found that in this particular field too, I believe. You took isolates and checked them, and
Yep.
sure enough, they're able to overcome that 1K gene, so that was not effective. But this really drives home the potential value, and we know that there's been lots of work, and Dorrance has done some great work looking at this as well, demonstrating that field tolerance is also a very important thing. This is much harder to breed for because it's a whole bunch of little genes that contribute to this field tolerance, but you can see really dramatic differences here, as you look down the line there between something with a good field tolerance and something with a poor field tolerance. The other piece of information that I think was really interesting, unfortunately, in this seed catalog, both of these varieties had the same field tolerance score. We called and we were able to get some information from the agronomist from that company, and they knew, unfortunately, that there were differences. It just hadn't made its way into the seed catalog. And this just stresses the importance of us all working together, working with private industry and our public breeders here to make sure that we provide the best information, not only on RPS genes that are effective and not, but also in terms of trying to screen varieties and understanding which have better field tolerance than other varieties. And here's an aerial shot here. And I've got to be careful with the yield numbers because, of course, these are two genetically different soybean lines. So they've got different yield potential. But as an example, if we assume that Phytophthora was driving a lot of this difference, we were seeing a difference of 60 versus 67 bushels to the acre at harvest. So that's, today's market in the order of $100 per acre difference, just based on which seed variety you selected, and that pressure from the Phytophthora sojae. So again, just trying to emphasize the importance here of, of course, we always look at yield data, right? That's important. Of course, it's the bottom line, but we also have to be protective of that yield and understanding which disease issues we have, and making sure we have a strategy in place by selecting varieties with good tolerance or resistance to these diseases is absolutely critical. All right, let's switch gears a little bit, Austin, and just real briefly. So Zach was another former PhD student in the lab now, and he pioneered this particular technique that Austin had to adapt, right, for sojae, and it's a little bit different. Zach developed it and used it more for Pythium species, and there's a whole separate talk on that. But Austin, tell us what you did here with this particular assay.
Yeah, so this is a high throughput assay for fungicide sensitivity screening. So traditionally, what we would do is make up a bunch of petri dishes that have varying concentrations of fungicides in them and test our isolates against them and determine kind of like a sensitivity number to each chemistry to determine if the isolates are resistant or insensitive. That takes a lot of plates and a lot of media and a lot of time to produce all the plates, transfer everything over, and then take the results down. So what Zach did was he developed an assay that uses these small 96 well plates that you can essentially do the same thing, but with many isolates at one time and using a lot less space and resources. Just to a brief workflow through this, we would grow these isolates on a very gel-like media that was easily macerated by pushing auger plugs through the syringe, and then we would use that macerate to inoculate these 96 well plates that each have a different fungicide concentration in them. So when you're looking at the plates, each well would have a different isolate in it, or there might be multiple wells with the same isolate in them for testing.
Mhm.
Um, and what we are able to do is, over the course of 24 or 48 hours, we can use what's called a spectrophotometer, or it basically shines a light down into those wells and determines how dense the organism is that's growing in there. And so when we have low concentrations of the fungicide, we have a lot of growth. Whereas when we have higher concentrations of the fungicide, if the isolate's insensitive, we'll have far less growth.
Mhm.
We can use that information then to go through and determine an EC50, which means that the effective concentration at which 50% of growth is inhibited. And that's typically the gold standard for looking at fungicide insensitivity or sensitivity.
Mhm.
And so by doing this, we were able to go through and screen all of our isolates much faster and with less resources than we would if we had gone through and done like a petri dish plate.
But just this is a great example, right? In the red, you've got an isolate that's able to grow at that 1 microgram per mil of that methanoxam, so about one
Hmm.
part per million, right, Austin?
Yeah.
There's some reduction, but it's still growing quite happily. And this other isolate here is inhibited at that same level. So that's just a visual representation of what's happening in a little dish that that's basically the size of your hand. So yeah, very nice technique. And what was exciting to me, Austin, is that you were able to get through 4 different fungicide chemistries pretty
Yes.
rapidly with your pool of isolates, right?
Yeah. Yeah, we were. So we tested 4 different chemistries. We tested 3 that are really tailored towards omycetes and are very effective on them, such as mefenoxam, ethoboxam, and oxythiopyprolin. Each one of these has a different frat code, which was also nice to work with. We
Yeah,
also sorry, go
sorry,
ahead.
I'll jump in real quick. I mean, the exciting thing there is too, like we're not mefenoxam's been around for a long time, right? It
Yep.
was about 45 years now. I think it was, registered in 1977. Um, so that's it targets these RNA polymerase inhibitors. So that's why it's given a specific frat code, right? It's just like thinking about herbicides. They have different mode of action and different codes associated. So what's really exciting is that we now have other products that have recently come to market that target different cellular processes. And so we're not likely to see cross resistance between the two. So if, for
Yep.
example, we do see resistance to mefenoxam, we should hopefully be able to control that pathogen still with these other modes of action that we have. And it will potentially put less pressure on that pathogen too, if we're using 2 different modes of action to treat that seed for protection.
Yep. We also tested one of the strobilurins, pyraclostrobin, because there's been some evidence that that is efficacious against Phytophthora sojay. So we just wanted to look into it as well.
Yeah, and it's widely used as a part of a seed treatment.
Yeah. Yep.
Mhm. Fantastic. So so I think these plots are really pretty to a scientist, right? But they're called violin plots. That's correct, isn't it, Austin?
Yes. Yeah. Yeah. They're violin plots.
Which like sort of who cares, but it's helpful for visualizing the data that you found, right? And Austin, do you want to walk us through what you saw and what this graphic means for these different chemistries?
Yeah. I'll start by explaining the graphic and then we can get into what it means.
Yeah.
So each one of these violin plots is for one of our 4 tested chemistries, and you can see those on the x-axis down there at the bottom. On the y-axis, we have the average absolute EC50. So this is for each isolate, the average concentration that we needed to inhibit 50% of the organism's growth. The violin plots themselves have a box plot in them, so you can see the median and then a general range of of data points. But what we're seeing here is that our older chemistry, mefenoxam, is still appears to be very effective against Phytophthora sojei, and that newer chemistries like ethoboxam are also very efficacious against Phytophthora sojei. So we weren't able to identify any sort of fungicide resistance to the compounds we screened. Pyraclostropin, also efficacious, has a little bit of a wider range there for data points. But still, if you look over on the y-axis, the highest data point is 0.2 ppm, which is very low. Oxythiopyprolin looks like it's disappeared, but it hasn't. Oxythiopyprolin was very efficacious against Phytophthora sojei, which is something we expected based on previous testing of other Phytophthora species. But oxythiopyprolin here, even the highest data point is barely registering on our graphic. So our lowest data point that we would be able to see is still below 0.02 ppm, which is very low.
Yeah.
We need less of that compound to inhibit 50% of the growth. It doesn't necessarily mean that it's a better overall product. And what I mean by that is we know that there are some inherently resistant species to both oxythiopyprolin as well as ethoboxam. So if we were just treating soybean seed with one of those, we'd control Phytophthora sojei great, but there are some other Oomycete species that would not be controlled by those compounds.
Yeah, that's a really good point, Austin. And the other point I would make too is that yes, absolutely. In this particular assay, oxythiopyprolin was good at inhibiting growth in this artificial condition, right? But there's a lot more to seed treatments than this artificial system. This is really important because it does certainly give us a current snapshot in time of how sensitive things are. We can look again in 5 years and see, has there been any shift? Is there anything to be concerned about? But the way the chemistries move in the plant, the way they bind, their solubility, all these other factors does certainly influence their concentration on the plant and the amount that you apply to a seed treatment. So there's a lot more that goes into the efficacy side of things as well. But this particular lab technique is really important for monitoring for fungicide resistance. Just like we need to be monitoring for herbicide resistance in our weed populations, we need to be doing the same thing here with our pathogens. So I'm really excited that you're able to generate this data. All right. So here's our overview point, I guess, right? Our action points. So I think Austin's made a pretty good case here that at least here in Michigan, RPS 1C and 1K generally offer little protection. Most of the isolates are able to overcome those widely deployed genes, unfortunately. There are genes such as 3A that's out there in quite a bit of germplasm now that did much better and is able to control more than 60% of isolates. And unfortunately, the pathogen is constantly changing, and now it's able to overcome more genes than it had 20 years ago. So we're stressing here that yes, RPS genes are still an important piece of management. We also need to be thinking about that field tolerance when we're selecting varieties. And just as a quick note, again, here's our RPS gene listing, right? Again, you can see most products that are offered tend to have that 1K gene, which again, we found not to be very effective. There are some with 3A, but we do have that information most of the time on the field tolerance of these varieties. So again, make sure you pay attention to that. And I'd also say, talk to your seed dealer. If you know you have an issue with Phytophthora, talk to your seed dealer and make sure you have the latest information, because these books are printed potentially a little bit ahead of when additional data might be being collected. So it's always important to have a good relationship and discussion with your seed dealer on that. And it was really nice to see that Austin wasn't able to find fungicide resistance that we were very concerned about. Obviously, we're going to continue paying attention to that, but there was nothing that we were, very concerned with at this particular point in time. So Austin, this brings me to the next part, right? What are you currently doing? This is really, yeah, to me, it's really exciting, but can you explain this to people?
Yeah, so I've gone through and looked at Michigan's Phytophthora sojei population and described effective and ineffective resistance genes. So I was interested in expanding that to the United States as a whole, as well as a few other countries. Um, so what I'm doing here is we are taking the last 30 years of these pathotype survey datas from individual states in the US, individual provinces in Canada, states in Argentina and China, and we're looking at that data to kind of identify when and where do these resistance genes efficacies start to wane and not become effective. So a lot of this is, reading the reports and the papers and taking that data down in a usable format so that I can look at it all together.
So this
So
is another thing that's taken me quite a bit of time, right? It's
yeah,
not time out in the field or grinding soil, which is kind of fun in itself, but
yeah,
this is
it's
computer time, right?
yeah, it's a lot less thrilling than being in a field diagnosing plants. That for sure. So I was able to amass 20 plus studies from the United States, Argentina, Canada, and China, which ended up being over 5,000 isolates data total. And we've taken all of these studies and kind of bend them into different time frames. But generally, we were using like a 1990s time point, so 90 to 99, um, early to mid 2010s, so 2000 to 2013, and then a more present day time point, which has been like the last round of pathotype surveys that were conducted in each country.
So in the US, you were able to find quite a bit of information, right? So you had more than pretty much 1,000 isolates for each of those, essentially the decades, right? The
Yeah,
90s, thousands, and the 2010s, which is really nice.
yeah, yeah. We were able to get great representation from the US from pretty much all of the major soybean producing states so that we could look at this. Um, Data from other countries was a little more sparse, and we didn't necessarily get as like a big of a data set as we did for the US. But we can still look at those countries. So we did a lot of the same stuff. We looked at effective resistance genes and we looked at pathotype complexity, same as we did in Michigan, but now on a grander scale. So just a reminder, pathotype complexity is the number of RPS genes an isolate can cause disease on. And what we're seeing here in these graphics, there's one for each country, Argentina, Canada, China, and the US. And then for each time point, we're looking at the average path or the spread of pathotype complexities for each time point in each country. And what we are seeing is that over time, the pathotype complexity is significantly increasing on this national scale. So, kind of backing up what we observed in Michigan is not just a Michigan specific thing. This is something we're seeing over all of the soybean acreage in the US, which was really interesting. And those trends hold true to the other countries too that we looked at, which again, interesting. This isn't just an isolated thing that's happening in the United States or even in Michigan. This seems to be a common realization.
Mm.
Likewise, similar story with the RPS gene efficacy over time. Same as we found in Michigan, we're seeing that RPS1C and RPS1K are no longer effective in the United States, Canada, and Argentina, while RPS3A does appear to be effective in the United States, Canada, Argentina, and China. China was kind of an outlier here. Phytophthora sojei is actually an invasive pest there, so it hasn't been around as long as it has here in the United States and Argentina and Canada. Um, so their data looked a little different, and we're still looking into that, trying to understand why that is. Um, yeah.
Mm. So what would you say the action points there? Like, I guess, across the US and, somewhat globally as well. Like what were you seeing, I guess, as a summary?
Um, so we were seeing that RPS 1C and 1K were no longer effective in three of the countries that we were looking at. So
Mhm.
we really need to start not just Michigan itself, right? We need to start as a country start looking at other resistance genes to deploy for management. Um, yeah, we also, I mean, same story we had with Michigan. The complexity is increasing, so it's less likely that we're going to be able to manage these populations over time with a single resistance gene. We may need more than one, and we will need to start looking more at partial resistance, field tolerance to help manage Phytophthora root rot in going forward.
Mhm.
We did identify RPS3A as a gene that was tested that we think would be a candidate for deployment to effectively manage Phytophthora root rot. There are a couple other RPS genes that have been reported and tested that we couldn't use. So things like RPS11 or RPS12, I believe, these appear to be widely effective resistance genes that companies are looking at introgressing into their lines. But so far, we haven't seen any varieties become available that contain those RPS
Yeah,
genes.
I guess that's a piece of excitement too, right? So it's a little bit doom and gloom data, right? Like,
Yeah,
unfortunately, like there's erosion of these genes, but we need to know that. And this is great information, for the public soybean breeders, but also the private companies. Like, they need to understand that this situation. And yeah, thankfully there have been the discovery of some new RPS resistance genes, and yeah, that's exciting, and we need to try and potentially get those deployed. And I guess I'd also stress as well, just making sure that we have the best information available to farmers. Again, yield drives everything. We know that, but if there's particular disease issues, we need to also be thinking about and managing for those, so we don't have these losses. So I would just like to say thank you very much. Some of this project actually started through the North Central Soybean Research Program, a grant that Ann Dorrance had pulled together. Some support here, Project Green, and of course, Michigan Soybean Committee, who are very supportive of the work we do. So thank you very much to those people that have made this work possible.
yes,
Um,
thank you.
yeah, and we know that this isn't live, this is recorded, but if you would like to reach out to Austin or myself, you can look us up pretty easily, online, but feel free to use Twitter as well. Reach out to us there and pose your questions if you have them. So thank you very much.